MarkUs Amann we're delighted to have him here. He's been here at the university almost as long as me, and we just haven't crossed paths before. So I'm really excited to hear about his work and everything that's going on here. By way of introduction, comes from Germany originally, did his undergraduate and master's degree at the Technical University of Munich. Afterwards, he came to the University of Utah for his PhD studies in integrated physiology. Yeah, exercise physiology was okay for the undergraduate and master's work as well. Exercise physiology.
No, just
oh, okay. Sorry, PhD, exercise physiology. He then went to the University of Wisconsin as a postdoctoral fellow for a few years. After that, accepted a faculty position in Switzerland for a couple of years before returning to the University of Utah and came here in 2009. He's currently a professor of anesthesiology, the Theodore H. Stanley Presidential Endowed Chair of Anesthesiology. He has adjunct appointments with the Department of Internal Medicine and Biomedical Engineering, and kind of has two worlds he works in here at the university and also at the VA with the GREC. Has grants both through merit program at the VA and here at the University of Utah NIH funding. On a side note, he's coming to speak to us on his birthday, so we're excited to have him here. Dr. Shah is celebrating his birthday. He's got some cupcakes to share.
Yeah, they're kosher if anybody's worried.
Worried about that.
Yeah.
And glad to have Dr. Amann on his birthday coming here with us. Robin, are you gonna lead us?
I'm starting. You guys jump in.
Happy birthday to you. Happy birthday to you. Happy birthday to Amann. Happy birthday to you. Thank you. I don't know that CVRTI has a choir going or anything, but excited to have him here. With that, we're excited to hear about your research and work, and we'll turn the time over to you.
Yeah. Thank you very much for that. That's a first for me to hear a song being presented to the presenter of this seminar. It's awesome. Thank you very much. Also, thank you, Robin, for inviting me, and thank you for you guys to come out and listen to my research over the last many years now, sort of a summary. I will be talking about neurocirculatory control mechanisms today, and the consequences of those for oxygen supply in health and disease. And the neurocirculatory control mechanisms, which are the mechanisms that control the cardiovascular response to physical activity, can in general be separated into two categories. We have a feed-forward mechanism and a pre-feedback mechanism. What these mechanisms do is they all have an influence on the medulla, on the cardiovascular control areas in the brainstem. And once activated, this results in changes in the autonomic nervous system. Parasympathetic outflow goes up or down depending on the mechanism, and sympathetic outflow also changes. And these changes have an effect on cardiac output, both on heart rate and stroke volume, and vascular conductance. And these two things in combination, three things actually, in combination, they have a hemodynamic consequence in animals and in humans. And that's basically the end point of these neurocirculatory control mechanisms. The starting point is right here. I start with central command. So central command is the one feed-forward mechanism, and that is associated with any physical activity that you do, whether this is just climbing stairs or going for a run. outside or just raising your hand. When this happens, there is a corollary discharge happening that activates the cardiovascular control areas here, and then this is the consequence. Another mechanism is the arterial chemoreflex, which has receptors in the carotid body right here or in the aortic arch. Once activated, feedback goes back to the medulla and then changes in sympathetic parasympathetic outflow. Very similar to this is the baroreflex, which is actually a negative feedback reflex. Once this is activated, parasympathetic goes up and not down as indicated here, and the sympathetic outflow goes down, but that's another reflex mechanism. And the last one, which is the one that I will be focusing on today, is the exercise pressor reflex. The exercise pressor reflex originates in the contracting skeletal muscle in the periphery. I'm depicting the muscle in the legs here right now. Once activated, feedback goes to the medulla and changes in the parasympathetic and sympathetic nervous system occur. This is an outline of what I will be trying to go through today. Ultimately, I want to land how the exercise pressor reflex can be part of the pathophysiology in heart failure. But before we get there, I would like to explain to you how it actually works in young, healthy individuals. And then does healthy aging from a 20-year-old to a 70-year-old, does this by itself, despite the absence of any disease, affect the function of this reflex mechanism? And then finally, the question is, does heart failure alter this reflex loop, and does this account for the patient's functional impairments that we all know about? The afferent arm of the exercise pressor reflex consists of group 3-4 muscle afferents. Those are sensory neurons which innervate skeletal muscle, and once activated, they project to various sites within the central nervous system. And here's a more detailed description. There are two types. There's group 3 and group 4. This is group 3. They are thinly myelinated sensory neurons. They have a conduction velocity which is relatively quick, 2.5 to 30 meters per second, and they are sensitive to mechanical stimuli, whether this is stretch or pressure within the muscle. Those activate these group 3 muscle afferents. And then there's group 4 muscle afferents. They are unmyelinated. They have a much slower conduction velocity, and they are sensitive to metabolites that accumulate as a result of muscle contraction. And the response of these was nicely documented 27, a long time ago, 97, sort of a long time ago, I guess. This is from Mark Kaufman's lab, where they had a dicerebral cat on a treadmill, which they stimulated electrically some areas in the brain, locomotor areas, and made that cat walk very slow. And at the same time, they recorded directly from group 3 and group 4 muscle afferents. And at rest, as you can see here, there was very little discharge, both group mechanosensitive and metabosensitive afferents. But as soon as the cat started walking, this is a very complicated preparation, so you cannot have that animal do exercise for a very long time. So they were able to get a minute out of it. As soon as the cat started walking, the discharge frequency of group 3 muscle afferents, the mechanosensitive ones, went up. And right at the end of exercise, the cat stopped walking, it stopped firing. Those fibers stopped firing again. And very similar with the metabosensitive ones, they were a little bit more noisy at rest here, but with exercise, they increased discharge all the way until the exercise was done again after a minute. Now, this is not a very drastic increase, I understand that, but please realize that this is literally a cat walking exactly the speed I'm doing right now, very slow. So with a higher speed, with greater intensity, the discharge would of course be much bigger. Now, the group 3-4 muscle afferents, they play various roles in a human, and I will be focusing a little bit on the role they have on the control of breathing, and the control of the cardiovascular system. I will not be focusing on their influence on central nervous system fatigue today. The question is, how do you study this? And my work is almost exclusively 99.5% in humans. So you have to think about how can I manipulate these group 3-form muscle afferents in a human, and there are different ways. You can use a technique called post-exercise circulatory occlusion, or PICO, which right here you see a subject laying there doing hand grip exercise, and you see a little cuff here right around the arm. And once you reach the end of the hand grip exercise, you inflate this cuff here. The exercise has now stopped, and you trap the metabolites within that muscle, and they continue to activate those metabosensitive muscle afferents. The discharge frequency remains elevated throughout the rest of the experiment. And that's a nice way to focus on the metabol reflex, or the metabosensitive reflex side of the exercise pressor reflex. That's the good thing. The bad thing is that this is happening at rest, so other neurocirculatory control mechanisms are not activated anymore, and there are big interactions between those, and that's completely neglected when you do this at rest with PICO. One of the results is that you overestimate the role of the metabol reflex. For example, central command is known to inhibit the dorsal horn neurons, which project this afferent feedback to the central, higher up to the higher brain areas, and that influence is gone. And you only focus on a subset of metabosensitive afferents, and I want to briefly show you what I mean by this. This is work done by Al Light in the Department of Anesthesiology a few years ago. He collected dorsal root ganglion neurons from mice, and he exposed them in a dish to different metabolite concentrations, mimicking rest, moderate intensity, heavy intensity exercise, and then ischemic exercise over this PICO post-exercise occlusion that I just showed you. The way he did it, he changed the pH and the ATP concentration and the lactate concentration. And what he found is when he exposed those DRG neurons, they responded in a certain way. As you can see here, the increase at rest relatively small, and then it increases to about the onset of heavy exercise, but then once the metabolites became more, they actually decrease the discharge frequency. And when you do the PICO technique, you are right here. So you're activating sensory neurons which are not even active or not very much active during physical activity. And then he found another subset of metabolites, and I just realized I just said this wrong. So this is actually, the gray ones are actually sensory neurons that respond during normal exercise, but once you get to these abnormal levels of metabolites, they stop firing. And this is the area where you do the PICO technique, so you are not even focusing on what's active during exercise. The other subset is relatively, the responsiveness is relatively low at these normal levels of exercise, but once you get to these extremely high metabolite concentrations, they start firing. Important thing is they are not active during physical activity. So that is the problem with the PICO technique. You're studying a subset of afferents which are not actually activated during physical activity. And then you can look at the mechanosensitive aspects of the exercise pressor reflex. And what we do here is we have a person sit in this chair and we passively move the leg. So we stretch the leg and extend. So we pull the leg out and back, and that stretches the muscle, that activates mechanosensitive afferents. The good thing is you focus on that. The bad thing, again, is it is at rest and it overestimates the contribution of the mechanosensitive afferents. So we weren't very happy with this, and then years ago, I think this was in 2007 or 8, we developed a different method of studying this, and we are now manipulating muscle afferents during voluntary exercise, muscle afferent feedback from the lower extremities, using lumbar intrathecal fentanyl. So that's an opioid, a new opioid receptor agonist that we inject right here at the lower back into the intrathecal space of the participant. So you go all the way in, you wait until a little bit of cerebral spinal fluid comes out, and then you put the fentanyl in. And that reduces the activity of 3-4 muscle membranes. And the good thing is you can do this during voluntary exercise, and the motor function remain completely intact. So the person is still able to walk, run, everything. It's just the feedback is attenuated to a certain degree. The bad thing with that technique is that it can facilitate the development of peripheral neuromuscular fatigue during exercise. The other not so good thing is that it only blocks about 50 to 60% of group 3-4 muscle afferents. It took us a while to figure that out, but it's a little bit more than half of the afferent feedback that we can block with this technique. Anyway, the rest of the talk, all the studies that I show you today is based on this approach. So first question is, how does this exercise pressor reflex, one of those neurocirculatory control mechanisms, work in a young, healthy person where everything is new and supposed to work as it works as it's supposed to work? And for that, we got young people. I forgot the age, but they were young, and we had them in this apparatus here, which is a knee extensor ergometer. What this allows you to do is it allows you to put the person on that seat, and behind that person there is a bicycle, and your right leg is connected to that bicycle, and you can exercise just the quadriceps muscle, a relatively small muscle mass, and you can perform physical activity there, and it allows you to do all sorts of measurements during that. What we have here is this is an femoral arterial and venous line, so we are able to draw blood samples from there. We quantify the blood flow in the femoral artery, and that allows us to use the direct Fick equation and calculate oxygen consumption of the and transport of the lower limbs, in this case the quadriceps muscle when they exercise. Yeah, I think I said everything I need to say. Yeah, so that was the methodology that we used for that. And here is the first finding. So this is femoral arterial blood flow measured with Doppler ultrasound. This is rest, control condition, fentanyl condition, and then these are the 3 exercise intensities separated in control and fentanyl on each of these. There was not an effect on peripheral blood flow at rest, but once people started exercising, as you can see, the blood flow response to this knee extension exercise was in almost all of the individuals that we tested significantly lower compared to that under control condition when lower with fentanyl when the group 3-4 muscle afferents were blocked. And as a matter of fact, there was a 15% reduction in limb blood flow, which is, when you're familiar with that area of hemodynamic responses to exercise, is a huge deal. It's huge. And other responses that we observed was the mean arterial pressure here and the venous pressure. Here are the workloads again. Mean arterial pressure was consistently lower during the physical activity in the condition with the blocked afferents compared to the control condition. And interestingly, also central hemodynamics. This is cardiac output, no effect at rest, but then during physical activity was significantly lower at all exercise intensities. This was mainly, it was a combination of somewhat of an effect on heart rate that was reduced, and then stroke volume. The interesting thing is, as you can see from rest to exercise, there is an increase under normal conditions with intact afferent feedback, but with the blocked feedback, there's really no change from rest to exercise. Stroke volume remains exactly the same. These two things combine, of course, to result in this reduction in cardiac output. So blockade caused a 25% decrease in central hemodynamics. This is just a brief excursion into the control of breathing, which is a whole other area that we study. But the interesting thing was there and this is actually not knee extension exercise, this is now exercising on a bicycle ergometer at different intensities. This one here evokes about 100% of VO2 max in these people. And again, placebo condition in solid green and fentanyl in the open symbols there. And as you can see, the ventilatory response relative to the metabolic rate during exercise without affecting it at rest was significantly lower during the physical activity. So drastic hypoventilation was happening. And this hypoventilation resulted in an increase in the end-tidal CO2, which reflects arterial CO2. And this is a drastic difference here from the control condition to the condition where people did the exercise with blocked afferents. This is basically what you would see in a patient with COPD with a severe lung disease. They retain CO2. This is what happened with the guys when we interrupted the feedback mechanism and the feedback contribution to the control of breathing. So a big effect. And this resulted, this is hemoglobin saturation, so saturation of hemoglobin with oxygen. There was nothing happening at first, but then later on, a big difference in the hemoglobin saturation during physical activity. And this hypercapnia that you can see here by itself is a very potent stimulus for breathing. And so when you factor this in, then our conclusion was that the blockade caused up to 50% decrease in the minute ventilation, in the ventilatory response, and consequently a substantial decrease in arterial oxygenation during physical activity. So point being, these afferents also play a huge role in the pulmonary response to physical activity. So given all that, there is potentially an effect on oxygen transport to the working muscle, which is absolutely needed to make sure that the fatigue resistance in a person is relatively high. So the question was, in these young, healthy people, what was the impact of these consequences of blocking the muscle afferents on the development of fatigue during physical activity? And so we did another study where we had them exercise. This is bike exercise again, and we measured the strength of the quadriceps muscle with electrical stimulation of the femoral motor nerve before exercise and after exercise. And under control condition, after exercise, this was 7.5 minutes or almost 8 minutes at a very high workload. After the exercise, the quadriceps was about 30% weaker compared to before the exercise. So exercise-induced fatigue. Now, when they repeated the exact same exercise, but with blocked muscle afferents, remember where the blood flow is down and the arterial oxygen content is down because of a reduction in hemoglobin saturation, what we found is there was substantially more peripheral fatigue in these individuals compared to the control condition. So in summary, on the healthy, what do these muscle afferents do in healthy young people? Well, blocking skeletal muscle afferent feedback compromises the hemodynamic response to exercise and impairs muscle O2 transport. And this is because of a reduction in arterial oxygen content and the reduction in lymph blood flow. And the conclusion there is, in young, healthy individuals, the exercise pressor reflex facilitates optimal O2 transport and peripheral fatigue resistance during physical activity. So it's a very important mechanism that we have to have in place in order to minimize the development of fatigue during physical activity. Now, the question then was, this is how it works in a young person. Does healthy aging affect the influence of these group 3-4 afferents and the pressor reflex on the cardiovascular response to exercise? So we studied older people, and the rationale for this was that previous studies have also already done hand grip exercise with that PICO approach that I showed you earlier, which we don't like very much. And they have suggested that aging actually attenuates the exercise pressor reflex. Again, they use a method that we don't think is ideal to study this. And one other thing that you need to consider when you deal with older individuals and compare them to young people is that there is age-related shifts in muscle fiber type, and those shifts are associated with different accumulations of metabolites, different kinds of accumulation of intramuscular metabolites in physical activity. And therefore, consequently, the difference in the pressor reflex might simply just be due to a reduced stimulation of these metabosensitive afferents. And so we thought it's time to look at this again. And that's exactly what we did again with this exercise modality here. Same one here, but now we had old people and young people. Again, arterial and venous blood samples and blood flow measurements during the exercise. And I think I forgot this earlier. This says here CO2 sensitivity test. So when you inject the fentanyl into the cerebral spinal fluid, there is a chance that it migrates from the injection site all the way to the brain. And there are opioid receptors all over the brain. But the nice thing sort of is once opioids make it actually to the brain, you have a ventilatory depression. The sensitivity to CO2 is down. And this is a good way for us to test whether the fentanyl that we inject here actually makes it to the brain. And if the CO2 sensitivity is down, then we know we cannot continue with that subject because any effect that we see might not be just due to reducing the afferent feedback with a spinal block, but also due to a direct effect of the drug on opioid receptors in the brain. So we do this all the time, and when the CO2 sensitivity is down, or if it is down, that subject is done and will not be considered further for data analysis. And this is what we found. So in young people, I already showed you some of this, the same thing again in this study, mean arterial pressure here, the different exercise intensities. In the young people, no effect at rest, but during exercise, the mean arterial pressure, this is actually perfusion pressure, arterial pressure minus venous pressure, this is the muscle perfusion pressure, was significantly down, and it was the same in the old people. So that side of the exercise pressor reflex was actually not different between young and old. It was about a 10% decrease in the mean arterial pressure in both groups. Now, so conclusion based on just that, aging has no effect on the exercise pressor reflex, which would support the earlier findings. But we looked a little bit closer, and this is what we found. So here are some other hemodynamic variables that we quantified. This is cardiac output here. This is systemic vascular conductance. And again, these are the exercise intensities here. These are young people, and no effect at rest, but during the physical activity, there was a significant effect on cardiac output, which was lower with blocked muscle afferent feedback. And it was similar in the old people. There was also a significant effect, but that difference was much smaller. And this is systemic vascular conductance. Again, no effects at rest. During exercise, systemic vascular conductance was lower with the blocked muscle afferents compared to the intact. And in the old, it was actually significantly higher. So this is a summary. Cardiac output decreased significantly less in the old. There was a 5% decrease in the old versus a 17% decrease in the young people. And vascular conductance decreased in the young people by 12%, but increased in the old people by 5%. Now, question of, was this combination in terms of central and peripheral hemodynamics, what does this actually do to limb blood flow? Because limb blood flow is ultimately what matters for a physically active human, because that is the ultimate last step that brings oxygen to the muscle. What the muscle then does with it, whether it takes it up and utilizes it, is a different story. But this is a very important point here. What was the bottom line in terms of blood flow? And again, this is already what I showed you. The blood flow, the oxygen transport to the leg was actually down in the healthy young people. And we did not see an effect in the old people. So the exercise pressor reflex had no effect on limb blood flow in the old people. So the conclusion from all of this was the contribution of the exercise pressor reflex in regulating the mean arterial pressure during exercise remains unaltered by the aging process. So the effects on mean arterial pressure are the same in young and old. However, in the young people, the exercise pressure reflex contribution to the mean arterial pressure response is predominantly regulated by cardiac output. And in the old, the exercise pressure reflex contribution to mean arterial pressure is predominantly regulated via vasomotor activity, an increase in vascular resistance in the periphery. And this shift compromises peripheral hemodynamics in the old and may help explain the limited exercise-induced peripheral vasodilation that we often see in older people. And taken together, healthy aging might actually unfavorably alter the neurocirculatory control during physical activity. And this got us into the heart failure studies. And the question there was, does heart failure actually alter this reflex mechanism even more? And does this account for the patient's functional impairment? Now, heart failure has many functional impairments. The ones that we are interested in is the peripheral hemodynamic response to physical activity. And here's a study that we did years ago. This is again this knee extension exercise, and you can already see patients with heart failure do much lower, are able to exercise at much lower intensities and external resistance compared to control subjects. But we quantified limb blood flow here and mean arterial pressure down here. Black is controlled and white is patients with HFrEF. In this case, this is heart failure with reduced ejection fraction. And every single workload, leg blood flow and therefore oxygen transport to the working muscle is lower in heart failure patients compared to controls. And this might be partially explain the functional limitations that these people see. Question is, what's the mechanism that contributes to this reduction in peripheral hemodynamic response? And it's important to say because usually in our lab we see a relatively normal cardiac output response. So the heart is really not the limiting factor in these people during this type of exercise, at least. And yet, despite normal heart rate, the peripheral blood flow response is down. We are interested in what is causing this. So the hypothesis is that group 3-4 muscle afferent mediated muscle reflexes impair the circulatory response to physical activity in heart failure with reduced ejection fraction. And the rationale for this study comes from previous investigations, which we have now reproduced over the years. People have found excessive sympathoexcitation during exercise in heart failure, patients with heart failure with reduced ejection fraction. The way they have done this is they have asked heart failure patients and control subjects to perform hand grip exercise. And we have a needle, a very small needle in the peroneal nerve, which is the sympathetic nerve, and that allows us to quantify the discharge frequency of peripheral sympathetic nerves. And that's exactly what you see here during rhythmic hand grip exercise. Muscle sympathetic nervous activity right here in healthy person. It's relatively quiet at rest, but with exercise, the discharge frequency goes up, reflecting an increase in sympathetic outflow during physical activity. And this is heart failure patient. The discharge frequency is much higher in these people, so much greater sympathetic innervation associated with physical activity in the patients. And the interesting thing was, when they, after the exercise, after the hand grip exercise, at the end of the hand grip exercise, they inflated a cuff and they trapped the metabolites in there. They did the PICO approach. What they found is in healthy people, there was somewhat of a reduction in the discharge frequency. It didn't go back all the way to resting levels and it stayed elevated. But when you compare this to what happens with heart failure patients, there was hardly a reduction there. So the conclusion there was that the muscle reflex, in this case the metabol reflex, might play an important role in this excessive sympathoexcitation in these patients compared to controls. And there's some animal work done, and we looked a little bit into the gene expression in healthy humans using dorsal root ganglion neurons from deceased patients. it seems like that the group 3 afferents are sensitized. This is a purinergic P2X receptor in the DRG is upregulated, and group 4 is desensitized. This is a TRP1 receptor, which are metabosensitive. There's a downregulated in the dorsal root ganglion, and so we were in touch with a tissue bank, and they sent us dorsal root ganglions of patients, heart failure patients that died of heart failure and controls, and we were able to quantify mRNA levels and proteins of certain metabosensitive and mechanosensitive proteins or receptors. Anyway, the question was, is the reduced limb blood flow in heart failure due to the excessive sympathoexcitation secondary to an exaggerated muscle reflex? And do group 3-4 muscle afferents limit limb blood flow in heart failure patients? So these afferents, one reason why the blood flow response to physical activity is down in the patients. We did the same study, the same approach again. This time we had heart failure patients. Here, this is New York Heart 2 and 3. This is the left ventricular ejection fraction, 24 plus minus 4%. So that's this reduced ejection fraction. This is the workloads that we did. The control condition, fentanyl condition, the CO2 breathing again, and we measure cardiac output, blood pressure, and blood flow again in the patients and in the controls. And this is what we found. These are our heart failure patients here. There will be the controls here. These are the exercise intensities. And this is cardiac output, stroke volume, and heart rate. So the central hemodynamics, and as you can see, the afferent block resulted in a reduction in cardiac output, which was mainly predominantly mediated by reductions in stroke volume in the patients. And in contrast to that is the effects of the group 3-4 afferents in healthy controls. And as you can see there was no influence on cardiac output in the controls. And the conclusion there from this slide is that the blockade caused a 10% reduction in cardiac output in heart failure versus no effect in controls. So these afferents play some sort of a role in the cardiac response in heart failure patients, but not so much to age-matched healthy controls. This is an important word. It's age-matched. So we match them with age, and if we can, BMI and some sort of other things, but no effect in these guys. This is mean arterial pressure and vascular conductance. Again, mean arterial pressure down in the heart failures, and vascular conductance actually increased significantly. And in the healthy controls, mean arterial pressure down, and also an increase in vascular conductance, but that increase was less. As a matter of fact, the blockade caused a 5% decrease in perfusion pressure in both groups, heart failure and controls, but it caused a 25% increase in leg vascular conductance in the patients versus only a 7% increase in the controls. So this is one more slide here on this. This is venous norepinephrine and the norepinephrine spillover, which sort of reflects the involvement of the sympathetic nervous system. And as you can see here in the heart failure patients, when we did the exercise with the blood muscle afferents, both the norepinephrine spillover and the venous norepinephrine concentration was significantly down in the patients. And as a matter of fact, it caused a 22% decrease in norepinephrine spillover in the heart failure patients. Now, this is not a very good indicator of what's happening during exercise in terms of sympathetic outflow, but it doesn't get any better than this during this type of exercise. You cannot record directly from a sympathetic nerve during exercise that moves the whole body. And this is ultimately the important thing. Again, this is femoral blood flow in the heart failure patients. When we block these group 3-4 afferents, we actually saw a significant increase in leg blood flow during physical activity compared to the controls where we did not see an effect. So the conclusion comes later, as I keep forgetting. Another slide. This is perceived exertion and fatigue. This is also very important when you deal with a patient population, or is perceived exertion. And when you reduce the group 3-4 muscle afferents, during the physical activity, the rating of perceived exertion actually went down. So the exercise felt easier for these patients in the face of block group 3-4 muscle exercise. And the other thing, most likely the result of the increase in blood flow, was that the level of end exercise fatigue, quantified here as a reduction in twitch force of the quadriceps from before to after exercise, was significantly less when the exercise was performed with block muscle afferent feedback. So less taxing, if you will, in terms of the neuromuscular or the skeletal muscle. So what we are left with, clearly heart failure with reduced ejection fraction impairs the role of the group 3-4 mediated muscle reflex in regulating the cardiovascular response to physical activity. And what we are left with at the moment is it the afferent arm of the reflex loop, i.e. is it the sensitivity of the group 3-4 muscle afferents that is affected by heart failure? Or is it an altered central integration of afferent feedback once it reaches the medulla and all the processes in there, which is a black box when you deal with studies in intact humans? That could be abnormal, we don't know. Or is it the efferent arm of the reflex loop, i.e. the sensitivity of alpha adrenergic receptors? This is actually something that we are tackling right now to see the alpha adrenergic responsiveness in these patients compared to controls. And it's most likely a mix of all of this. Now, this is the summary. Muscle reflexes contribute to exercise intolerance in heart failure with reduced ejection fraction. They limit the peripheral hemodynamic response to exercise. They exacerbate the effort perception, and they exacerbate the development of fatigue, both peripheral and central. And I didn't go into distinguishing these two components of fatigue today, but it affects both. On the positive side, this is a good starting point for future pharmacological approaches. The reason why I'm saying this is, of course, we cannot have patients with heart failure, we cannot always give them spinal blocks if they want to do any sort of physical activity or get enrolled in a physical activity-based cardiac rehabilitation program. But if we could come up with a drug that somehow silences the feedback from these group 3-4 muscle afferents, feedback to the central nervous system, to the brain, that might help. And this is a potential target to ameliorate exercise intolerance in heart failure patients. And this might improve the success of cardiac rehabilitation, which is a very effective tool in managing heart failure. And if we could come up with some way of making a patient with heart failure be able to contribute, to participate a little bit more effective in a physical rehabilitation program, the success might be greater. So this is the last slide here. This is what I showed you today. I showed you when we block group 3-4 muscle afferents, what happens in healthy young people, in older people, and in heart failure, patients with heart failure. In healthy young people, we see drastic reductions in cardiac output and a reduction in systemic vascular conductance. And this combines to reduce limb blood flow in these young people. In healthy old people, these group 3-4 muscle afferents, they have no longer an effect on cardiac output probably because of the insensitivity of beta receptors in these people to sympathetic innervation. And we see an increase in systemic vascular conductance in these people. These two things combine to reduce mean arterial pressure and ultimately have no effect on limb blood flow and therefore limb O2 transport. In heart failure, we see somewhat of a moderate reduction in cardiac output and a huge increase in systemic vascular conductance. And that combines to reduce, again, mean arterial pressure, but importantly, it actually increases limb blood flow and O2 transport to the exercising limb muscle. And this is a slide from the people that I've worked with over the years and helped in different countries with these projects. And this is it. This is the end if you have questions. I'm ready.